This video will cover enzyme kinetics and reaction rate. We will look at the Michaelis-Menten model, key constants like Vmax, Km, and Kcat, and the linearization of the Michaelis-Menten equation. We will also discuss the phenomenon of cooperativity and allosteric enzymes. This scene lists the main topics we will cover: enzyme kinetics and reaction rate, measurement of the rate of an enzymatic reaction, the Michaelis-Menten model and its constants, linearization, cooperativity, and allosteric enzymes. What is enzyme kinetics, and why is it important to measure and understand it? We will answer these questions in the following scenes. Enzyme kinetics is the study of the rate of biochemical reactions catalyzed by an enzyme. It helps us understand enzymatic efficiency and determine enzyme concentration in a sample. Studying the mechanism of action of an enzyme is best done by determining its reaction rate and how it changes with experimental parameters. This is the core of enzyme kinetics. To summarize, enzyme kinetics is determining the rate of an enzyme-controlled reaction and studying how that rate changes. The reaction rate, V, is measured as the change in substrate concentration over time, or the change in product concentration over time. Enzyme activity is measured by observing the disappearance of the substrate, or the emergence of the product. This is done with very small quantities of the enzyme under optimal conditions and substrate saturation. This graph illustrates the typical course of an enzymatic reaction over time, showing the increase in product and the decrease in substrate concentration. In an enzymatic reaction, the formation of the product is proportional to the disappearance of the substrate. The reaction involves the formation of an enzyme-substrate complex, ES. The rate is defined by a steady state, where the concentration of ES is constant. The steady state is reached when the concentration of the enzyme-substrate complex, ES, remains constant. At this point, the rate of ES formation equals its rate of disappearance, allowing us to simplify the rate equation. The steady state is the assumption for applying the Michaelis-Menten equation. Enzymes that follow this pattern exhibit a hyperbolic relationship between initial velocity and substrate concentration. V0 is maximal when the enzyme is saturated. The Michaelis-Menten model is central to understanding enzyme kinetics. The Michaelis-Menten equation was developed by Leonor Michaelis and Maud Menten, who laid the foundation for the mathematical description of enzyme kinetics. The Michaelis-Menten model describes enzyme kinetics, where the graph of initial velocity V0 versus substrate concentration S is hyperbolic. Key parameters are Vmax, the maximum speed, and Km, the Michaelis constant. The Michaelis-Menten equation derives from the steps E plus S forms ES, which then forms E plus P. The initial speed and steady-state conditions lead to the hyperbolic Michaelis-Menten equation. This image highlights the hyperbolic curve of the Michaelis-Menten equation, showing V0 versus substrate concentration and illustrating Vmax, Km, and V0 over 2. Vmax is the theoretical maximum speed achieved when the enzyme is saturated with substrate. It is a constant and is never fully reached, as the V0 versus S curve is asymptotic. Km, the Michaelis constant, is the substrate concentration at which the reaction rate is half of Vmax. It ranges widely and reflects the enzyme's affinity for its substrate. Km is sensitive to variables like pH and temperature. Different enzymes have different Km values, indicating varying affinities. For example, hexokinase has a low Km for glucose, working well at low concentrations, while glucokinase has a high Km, functioning for storage at high concentrations. This image is a reminder of the hyperbolic curve defined by the Michaelis-Menten equation, essential for understanding Vmax and Km. The Michaelis-Menten equation explains experimental data. At very low substrate concentrations, V0 increases linearly. At very high concentrations, V0 approaches Vmax. When V0 equals half Vmax, the substrate concentration equals Km. Kcat, or the turnover number, is the number of substrate molecules converted to product per enzyme molecule per unit of time under saturating conditions. It is calculated as Vmax divided by the total enzyme concentration. To more accurately calculate Vmax and Km, the Michaelis-Menten equation can be linearized using the Lineweaver-Burk plot or double reciprocal plot. Plotting 1 over V0 versus 1 over substrate concentration yields a straight line. Some enzymes show a sigmoidal curve of V0 versus substrate concentration, indicating cooperativity. This means the binding of one substrate affects the binding of subsequent substrates, often seen in multimeric enzymes. For enzymes with cooperativity, the V0 versus S plot is sigmoidal, and Vmax over 2 is referred to as K0.5. Affinity increases as sites are occupied, unlike Michaelian enzymes. Cooperativity and allosteric effects are explained by two models, the Monod-Wyman-Changeau concerted model and the Koshland-Nemethy-Filmer sequential model. Both postulate enzyme subunits exist in low-affinity tense or high-affinity relaxed conformations. In the concerted model, binding of the first ligand shifts the equilibrium towards the relaxed state. The sequential model involves subunits changing conformations sequentially upon ligand binding. Allosteric enzymes possess a quaternary structure with multiple active sites, and their activity is regulated by allosteric modules, which bind at sites distinct from the active site. Allosterism is the property of proteins to undergo a conformational change upon ligand binding, affecting the affinity for that or another ligand. Allosteric effectors are the ligands that modulate enzyme activity. Allosteric enzymes can be regulated by both activators and inhibitors, which bind to the allosteric sites and modulate enzyme activity. Homotropic effectors bind to the active site, which also serves as the regulatory site. Binding at one site affects affinity at others, leading to positive or negative cooperativity. Heterotropic effectors are molecules that bind to a separate regulatory site, altering the enzyme substrate binding affinity. They can either activate or inhibit the enzyme, leading to positive or negative heterotropic effects. This image illustrates negative and positive heterotropic effects on enzyme activity, showing how different effectors modulate the curve of reaction velocity versus substrate concentration. Glycogen phosphorylase is regulated allosterically. Glucose and ATP, signaling high energy, shift the balance to the inactive tense form. AMP, signaling low energy, shifts the balance to the active relaxed form. Phosphofructokinase 1, PFK1, a glycolytic enzyme, is allosterically inhibited by citrate, which is not a substrate. This is an example of feedback inhibition, regulating the pathway. Aspartate transcarbamylase, ATCase, is inhibited by the end product CTP, stabilizing the T-shape, and activated by ATP, stabilizing the R-shape. This regulates pyrimidine nucleotide synthesis. The graph for ATCase shows that the activator ATP increases activity and sensitivity, while the inhibitor CTP decreases activity and sensitivity to substrate concentration. Protein-ligand interactions are specific and involve weak bonds, such as van der Waals, hydrogen bridges, hydrophobic, and ionic bonds. The functionality of proteins depends on flexibility and conformational changes. This slide marks the end of the video content.